All right, welcome back, everybody. I hope we're doing well. Hope we're hanging in there. What we're going to be doing in this lecture is spending a lot of time talking about not only how we examine the brain in neuroscience and biological psychology, but also what we know about it on at least the surface level. Much like our last class, we're not going to necessarily dive into all the nitty-gritty details of this specific topic. It's just impossible to do it in one or two lectures. But hopefully after leaving this specific class, you have a broad overview of some of the things that you expect to know when leaving this intro class, and it might help you better understand some of the more specific things that you might learn about in future classes that look at the brain and all the things connected to what we call the nervous system. Just so we have some context as to how we got to where we are today. If you recall in our last lecture, we spent a lot of time talking about a critical component to how our mind works, and it was a cell called the neuron. We talked about what their structure was, some of the different things that they did, and how they came together to allow us to do a lot of the elaborate things that our mind is capable of doing. But what we didn't necessarily spend a lot of time talking about was the structures that these specific neurons come together to actually make. That's what we're going to be looking at today. And this leads us to the discussion of the different levels of not only biology, but the mind that we can potentially study in the field of psychology. If you remember back to our second lecture, we talked about how some people in psychology might study the mind on the cellular level. We also talked about how some might study it on the brain level, or some might look at the organism and potentially even beyond when we get to things like social psychology and cultural psychology. Well, what we're going to be doing today is sort of looking at an area in between studying the mind on the cellular level and studying the person as a whole. Specifically, we're going to be talking a lot about the brain and all the things connected to it in this big system that we again call the nervous system. To understand the nervous system and the brain in particular, what we first have to do is really clearly define the two things that make up this nervous system and allow our mind and body to communicate with each other. There's lots of different ways that we can parse out the nervous system, but the primary way most people first learn about some of the breakups of the nervous system is when we differentiate between what's called the central nervous system and the peripheral nervous system. The central nervous is comprised of the brain, what's called the midbrain, hindbrain, and spinal cord. Now, when we think of the central nervous system, we tend to focus on that spongy structure on the top that's called the brain. You'll often also see it described as the neocortex. But underneath that neocortex are a bunch of subcortical structures that are critical for our not only day-to-day functioning, but really second-by-second functioning. And there's also other structures that are connected to those inner structures that are critical players in lots of more subtle things that are not only essential for human survival, but the survival of many, many species. We're going to spend a little bit of time talking about some of those other structures, but most of our attention is going to be turned to the brain, how it's kind of comprised, and some of the different things that you should be aware of when talking about the differentiation or allocation of specific aspects of the brain. It's also important to note, though, that that central nervous system really only works as a result of all the different nerves extending to different parts of the body. And these nerves that extend out to the body and the nerves that come from the body to eventually the brain, either via the spinal cord or through other paths that are semi-rare if we talk about things like cranial nerves, what we're talking about is this peripheral nervous system. Now, usually when we think of the peripheral nervous system, we think of it being linked to our sense of touch and our motor system. But there are other critical components to the peripheral nervous system that are important to note. Much like we talked about people tending to focus on the cerebral cortex or neocortex, or the spongy part on the top of our heads when talking about the brain, Most people do focus on the nerves extending to things like the skin and muscles that are critical for those motor and sensory systems, but we also have nerves that go to lots of our vital organs that are critical for our second-by-second functioning. And sort of recognizing those things is a big first step to understanding the importance and also complexity of this thing we call the nervous system. As I mentioned before, though, when we talk about the brain and how it works, especially in an intro class, most of our focus is on that sponge-like structure on the top of our heads that we call the cerebral cortex, or sometimes the neocortex. Now, if we look a little bit closer at this neocortex, what we can see is that even though there's lots of individual differences, in fact, almost no two people's brains look even remotely alike. Well, okay, that's an over-exaggeration. No two people's brains, I guess, are identical. What we see is, despite the kind of uniqueness of each individual's brains, there are some kind of breakups of structures that are not only observable by the time we're adults, but there are indications that these structural differences are even present at birth. A fun little note that you might want to be aware of as you're progressing throughout future classes is that our brain looks relatively smooth and simple when we're born. And this is really a byproduct of the lack of connections between neurons that we talked about earlier. In essence, we need lots of what we call synaptogenesis, connections between neurons in those synapses, and lots of supporting staff cells, like the glial cells we mentioned before, for the cerebral cortex to work as effectively as it does. And this is something that takes time. And as we age, as these connections start to grow, the surface space required for those connections to exist increases as well. And since our brains can't necessarily expand beyond our skulls, and since our skulls fuse at relatively young ages, the only path for these connections to be made is to create extra wrinkles in the brain, essentially allowing for extra surface space for these cells to connect. So as we age, certain things that are already present at birth become more apparent. One of the things that becomes more apparent is the existence of these things that we call lobes. Four general sections that we like to indicate have specialized functions attached to them. Now, if you do progress in a neuroscience class, you're going to learn that what we cover here in an intro class is extremely simplified, and the communication process and all the allocations of resources are much, much, much more complex than what we would cover in a class like this in terms of the rapid pace of action between different areas and the kind of diverse specification of specific areas of the brain that we don't get to in a class like this. But for the sake of simplification and kind of the critical things that you would want to know moving forward, we can say that your four lobes tend to be associated with four general things, even though two are definitely more diverse than some of the others. If we start with kind of the simplest lobe, even though it does a lot of very complex functions, we can start with the lobe that's in the back of our brains called the occipital lobe. Now, this occipital lobe is used for a number of different things, but its primary function can be sort of identified through another name that this occipital lobe is given. You'll often see it described in textbooks as the visual cortex. That sort of indicates exactly what most of the neurons and cells connected to those neurons are responsible for in terms of its kind of utility in the mind. Even though lots of other lobes do carry some of the load in terms of visual processing, the very first visual processing inside the cerebral cortex happens in this occipital lobe. And that information that's processed in the very back of our brains works its way forward as we start to break down all the complexities of the visual world that we encounter. One of the places that visual information goes to is the lobe located above and in front of the occipital lobe called the parietal lobe. It is true that the parietal lobe does play a role in visual processing, But if we want to simplify things again, we can talk about a critical component to the parietal lobe that's actually highlighted in the very front of this section. There's a band in the very front of the parietal lobe that we call the primary somatosensory cortex. And much like the visual cortex, the name of that band sort of gives away what this section is for. This area seems to be what we call projection area for a lot of the stimulation that occurs outside the body and is sensed to the brain and is recognized as some type of touch. Now, in your future section, you're going to learn about how this particular strand, or I guess strand of brain has another kind of interesting breakdown to it when we introduce ourselves to something called the somatosensory homunculus. Essentially a weird, distorted representation about the amount of space that we dedicate to different parts of our bodies along that primary somatosensory cortex. You'll also, in that section, spend a lot of time talking about the differentiation between the two different hemispheres of the parietal lobe. Another thing that is actually extremely distinguishable when we look at not only the brains of infants, but the brains of humans. And when we talk about hemispheres, what I'm alluding to is the thing that you see on the left picture. The fact that our brain, when seen from above, sort of looks like two relatively equal things, or two equal halves, that come together at a juncture that we call the corpus callosum. There's actually a couple other bands that tie our two hemispheres together, but the corpus callosum is the primary one. And that allows for messages from both sides of our parietal lobe and our occipital lobe and all of our other lobes to communicate with each other effectively. There's a really cool thing that's described in the book when people have this corpus callosum severed for a variety of different reasons that I encourage you to read about. But know that that pathway is there to allow both sides to communicate with each other, because both sides of our parietal lobe, and actually all of our lobes, are differentiated for different things. And we're talking about our parietal lobe and the primary somatosensory cortex. We talk about something called contralateral communication. The fact that the left side of our parietal lobe is actually responsible for processing all the stimulation that's occurring on the right side of our body. It's also true that the right side of our parietal lobe is responsible for processing all of the stimulation of things occurring on the left side of our body. And this process of contralateral communication also works for our motor system, which starts in an area right in front of the parietal lobe called the frontal lobe. There's a band, much like we have the primary somatosensory cortex in the parietal lobe, there's a band called the primary motor cortex that's found in the very back of the frontal lobe. And as you could probably expect, there's a lot of interplay between those two different areas, and there's actually a lot of overlap between them. But much like we have that contralateral communication in the parietal lobe, we also have it in our frontal lobe, where the left side of our frontal lobe starts the process of motor systems on the right side of our body, and the right side of our frontal lobe starts the processing of motor systems in the left side of our body. But it's also important to note that when we talk about the frontal lobe, we usually focus our attention not as much on the primary motor cortex, and instead turn our attention more to the area in the front of that frontal lobe called the prefrontal cortex. And the reason for this is multifaceted. I guess if we're looking at the biggest reason why we focus on the frontal lobe is because we believe that this is one of the areas that really distinguishes humans from most other species. We as humans have much larger prefrontal cortexes and much larger frontal lobes overall than almost every species. Well, actually than every species that we've encountered. And this area of our brain seems to be something that takes a long time to form, and as it forms, seems to correlate very highly with lots of what we call executive functioning. Things like impulse control and planning and organization of information seems to have a kind of a high level of overlap with activity in this prefrontal cortex. And it's probably not just random chance that as this area continues to develop, actually until our mid-20s usually, we continue to improve in these executive functioning skills. This brings me to one of my favorite stories, and I love to tell to students when we are in an in-person class. This notion that many of you, when you learn this, might feel inclined the next time you argue with your parents about why you didn't do something that you were supposed to do, you could tie in material from this class and explain this in kind of a basic neurological level. I always tell students who start to kind of go down the path of what they could say to their parents, that they have to be reminded of the fact that it's not that until 25 you don't have a prefrontal cortex and then magically it just pops in. It's there, it's building, and in fact, the more you work it, the more it will develop and the faster it will develop in your brains. But, it is kind of an interesting adage, and again, gives us some sort of food for thought when it comes to a lot of the different things that we learn about in this class. But it's important to note that, again, the frontal lobe is pretty multifaceted. It's not only responsible for a lot of these kind of higher order mental functions, but it also starts some motor processes, and you're going to learn about in your future sensory systems lecture that it plays a role also in lots of movement processes that involve coordination and other types of function. And we're looking for other complex lobes that we have in our brain. I've saved the most complex lobe for last. It's a lobe located below the parietal and frontal lobe called the temporal lobe. An easy one to remember, just think of it as located just below your temples. The temporal lobe sort of looks like an ear flap on the left and right sides of your corpus callosum. And this is by far the most complex lobe that we've identified in the brain. Well, it's not that we're going to identify more, it's just that exists in our brain. And this is also a lobe where we're talking about the different hemispheres becomes very complex and important. So some of you, before getting into this class, might have heard people describing themselves as very left-brained or right-brained, and you might have wondered at the time what the heck they were talking about. People who describe themselves as right-brained tend to think of themselves as very intuitive and artsy and creative, and I don't know, I always think of a flower child as the prime candidate of somebody describing themselves as right-brained, and somebody who's very logic and math-driven being described as very left-brained. Now, that's kind of an oversimplification of how these two sides work, but it is true that when we do engage in artistic activities and are trying to come up with new things in creative ways, what we see is a high degree of activity in the right side of our temporal lobe. And when we talk about language and logic, basic math, what we see is there's a high degree of activity in the left side of our temporal lobe when we're engaging in these things. And in fact, in a few slides up, we're going to talk about how we're sure that there are specific areas within the left side of our temporal lobe that are critical for language functioning when we talk about how we actually know this information about the brain. And that brings me to a really important topic, and that's the fact that, well, how do we know this? We've covered the basics, the stuff that you'll need to know in your future classes, but what I also love to talk about in this specific lecture is how we've come to conclude all of these amazing things about our central nervous system and how we might even know about that peripheral nervous system that we talked about earlier. And this brings me to the remainder of this class, spending some time examining how researchers have been able to study the brain in all of its complexities in the short time span that we've had to do it. As I mentioned in our last class, it wasn't until about 150 years ago that we got our first images of a neuron. And it wasn't until just recently, within the past 70 or 80 years, that we really had good tools to better understand the complexities of the brain. In fact, we're still improving a lot of these tools as we progress. So how do we get all of this information in that short span of time? It's probably first important to note that even before we were able to image neurons and study all the complexities of the brain in the way we can now, people were still tinkering with the brain in attempts to understand it. Most early researchers didn't necessarily focus their attention when trying to understand the brain on human brains, but a lot of them focused their attention on animals. Now, many of the things that they did hundreds or thousands of years ago to understand the brain in this method would be considered extremely unethical today. Lots of individuals would find animals off the street or find random things that they could dissect, cut their skulls open and tinker with the brain until these individuals would no longer be able to survive, and they would just write down what they were able to conclude in the short time span that these individuals were living. This gave us some broad senses of how the brain might function, but obviously there were lots of issues with this. I mean, ethics aside, which we should never throw aside, but ethics aside, even if we could find a way to do this in a humane way, this information was not necessarily as helpful as we maybe would like it to be. The ability to correlate the brain of a rat or mouse or monkey or some other animal with that of a human is folly, I guess. There's a lot of differences there, even if there are lots of cortical overlaps as well. There's also the inability to control for individual differences and all of the different nuances to each brain when you're trying to understand the brain on this surface level. But it was one of the first ways that we at least were able to determine the basic functioning of the brain. And it allowed us to determine something which was really critical that we call necessity. It allowed us to say, look, if you don't have this section of your brain, this function goes away. We don't know if other parts of the brain are maybe active when you're doing certain things, but you lose functionality if this is gone. Another type of study that allowed us to determine this was what we called human case studies. These were much, much more rare than animal research, but they gave us amazing insights when we were able to conduct them. The classic example of a human case study that more people cite than any other one in textbooks is the case of a gentleman named Phineas Gage. He was a railroad worker in the United States, actually, a little over a century and a half ago. Gage, when he worked at the rails, was in charge of a number of individuals, and according to the story that has been told over and over again in textbooks, he got distracted one day while laying dynamite and rods down to get the ties in the rails set. And apparently during this distraction, the piece of dynamite that he was using to shoot the ties down with his rods accidentally shot a rod up through his chin, through the back side of his his left eye, and up through his skull, when it was traveling at such a high speed that it actually cauterized the majority of the wound, and he was able to survive in a time where we definitely didn't have a lot of medical advances that we would consider essential today. Phineas Gage's ability to survive gave individuals in neuroscience and just science in general the ability to peer into what would happen to an individual if a large portion of the frontal lobe, I guess, of our brains was removed. There were lots of overblown stories of Phineas Gage being unable to control himself and only being able to handle life with animals, but his demeanor did change. He did shift. And our first inklings as to the importance of the prefrontal cortex came from cases like Phineas Gage. There are also other scientists that would not necessarily find people in these sensational cases, but people who had maybe suffered strokes and somehow survived, having portions of their brain sort of die off, knowing that they would not return, that would lose them specific types of functioning, at least for a short period of time. And these individuals that would experience these things would be tracked, they would have all of the different functions tested, and when they passed, researchers and scientists would dissect their brains and try to figure out which sections had the damage and what that indicated for the necessity of specific regions for specific types of function. One of my favorite examples of this is the example of what we call Wernicke's and Broca's aphasia. Many individuals who suffer damage to the left side of their temporal lobe lose the ability to communicate effectively. But what Carl Wernicke and Paul Broca discovered is that the type of language deficiency that people experience is highly dependent upon the area of the brain in this temporal lobe where the damage occurs. Wernicke's research led us to conclude that the production of language, the ability to create a coherent sentence or at least an idea that we wanted to convey, seemed to occur in what we call dorsal region of the temporal lobe. In fact, it also worked its way up just a little bit into the parietal lobe. Broca's area, which was much closer to the frontal lobe and actually did encompass a little bit of the frontal lobe in addition to this temporal lobe, seemed to be critical in the physical production of the language, the spoken language that was coming out of our mouth. And there were other areas very close to Broca's area that were critical in things like writing out the thoughts that we had in our mind. And the stark difference between these two areas is very easy to identify if we say we're able to watch videos of somebody struggling with what we call Wernicke's aphasia or Broca's aphasia. Wernicke's aphasia patients show a great deal of frustration when they're trying to have a conversation with somebody, and let me correct myself. Wernicke's aphasia patients don't show a lot of frustration when they're trying to communicate with somebody because they don't seem to identify, unless they're getting cues from the other person, that what they're saying, what they're doing doesn't make perfect sense. Broca's aphasia patients, on the other hand, show a great deal of frustration. They struggle constantly to be able to get ideas across, and those struggles manifest themselves in them repeating themselves or showing agitation when they're trying to produce something that they've formed in their minds. Now, if we were in an in-person class, we'd actually be able to watch 2 videos of individuals who had suffered from strokes and had these types of aphasia, but you'll learn about these things, I promise, more in future classes. And again, these case studies, these examples, really highlight how even before we were able to take images of the brain, even before we could look at neurons down at a cellular level, we still had a decent basic understanding of what sections of the brain you needed to do specific types of functions. And this is really the biggest plus of both animal case studies and human case studies. By waiting for people to essentially pass and looking at which regions of the brain they had damage to that lost them the function that they lost, well, we could say, you know what, if you don't have this, you can't do this type of function anymore, at least for a short period of time. With the animals, when certain areas of the brain were knocked out and they suddenly lost the ability to do grand functions, well, again, we can be pretty certain that that area probably was dedicated to something related to that specific action. But these particular approaches were extremely limited in other ways. There was, as I mentioned before, lots of lack of experimental control with animal research, and the same was true with human beings in case studies. Researchers couldn't say, what I'd really like you to do is have you have a stroke right in this one spot of your brain, or have a bullet or a rod shoot through this one spot of the brain, have you survive, and we'll see what you lose the ability to do. They just took who they got and looked at what they couldn't do, and oftentimes just sort of found general broad areas that seemed to be associated with specific types of function based on multiple people who seemed displayed to certain types of deficiencies that had damage to certain parts of their brain. Another thing, which is somewhat frustrating for researchers, though advantageous for humans and other animals, is that there's this thing called neuroplasticity. It's very helpful for us because we actually have the ability to regain lots of functioning even if we experience damage to certain parts of our brain, because other areas can sort of pick up the slack. It's not that we're usually perfectly fine if we have major damage to huge sections of our brain, but it is amazing as to how much we can regain specific types of mental functioning if we do have damage to certain parts of our brain, and how even if our brains develop in unique ways, neuroplasticity can allow many people to behave in a perfectly typical way, even without large areas of their brain. In fact, this brings me to a fun experience that you might have if you ever get to know a neuroscientist, where they'll take an image of your brain, point out some section that doesn't look perfect, because I promise all of us have brains that don't look perfect under a scanner. There's all these different random things that pop up that make our brains look nothing like the ones that we see in a lot of the pictures we've looked at so far. But those individuals who are joking and telling you that you're not going to be able to do things because you have certain parts of your brain are really, well, just taking advantage of our lack of understanding of how important neuroplasticity is for pretty much all humans. Most of our mental functioning is housed in the certain areas of the brain that we've talked about here. But there's always unique individual differences because all of our brains are slightly different from each other. And the reason why we're not incapable of doing something because we don't have a certain part of our brain is this neuroplasticity that we talked about. I guess it's also important to note before we move on that, with neuroplasticity and the lack of experimental control being a problem, another big issue with this being our only way of understanding the brain is when this was around as our only technique, it wasn't that often that people survived damage to the brain. So when we started to pick up in our scientific endeavors, when we started to try to understand the mind and the body in other ways, researchers quickly realized about 100 years ago that we had to find new techniques to help us understand the brain better. And this brings us to around the 1920s. At that point in time, researchers started to develop devices that could look for changes in electrical charges to different things, and came to the conclusion that we could actually look at brain activity by trying to detect these same electrical changes. Now, we didn't really understand what was the cause of these electrical changes at the time, but for those of you that remember back to our last lecture, you might remember that we talked about how when neurons are active, there's this switching or changing of electromagnetic energy at the axon level of each neuron. Well, certain detectors, very sensitive ones, can actually pick up on these changes if numerous cells are active in certain regions of the brain. And this led to the development of something called the electroencephalogram, or the EEG. You see kind of the basic version of an EEG pictured here in this slide, where they put a big, almost hat on a person with lots of sensors so we can get a general sense of where certain areas of the brain are active. This allowed us to kind of get a broad sense of which areas of the brain were active when we did specific functions, not necessary, but active when we did specific functions, and was able to measure these activities because of how sensitive these types of devices are down to the millisecond. This also led to our recognition that if electrical activity is involved in brain activity, well, we could maybe trigger specific things through just kind of replicating these electrical charges. This led to electrical stimulation studies that started in the 1930s and beyond, where researchers would actually go into the brains of individuals, trigger certain spots to see what would happen to an individual when these parts were triggered. A classic study that highlighted what we could maybe potentially do with this was a work of a now somewhat discredited researcher named Penfield, who swore in his research that he was able to identify what he called engrams of specific memories. His contention was that when he went into the hippocampus of individuals' brains, an area that we now know is definitely dedicated to memory, He could shock specific parts of that hippocampus and elicit specific memories within individuals. Nobody's been able to replicate Penfield's research, and what we seem to suggest now in our research is that memories are more a byproduct of neural activity, not specific neurons. But this type of research, these endeavors, triggered an onslaught of studies to try to figure out other ways to examine the brain, and also triggered an onslaught of things that we did even if we didn't understand what it was we were doing at the time. You might read about in future clinical classes an approach that became somewhat popular over 100 years ago called electroconvulsive shock treatment. This notion that if the brain is working on these electrical pulses, we might as well, if somebody's brain's not working well, just reset the whole thing by jolting somebody with large numbers of electromagnetic stimuli. Why in the world we'd think that would necessarily fix something is a little beyond me, but there's some debate about the utility of this in the past, and there's even some lingering approaches that don't necessarily try to reset the entire brain, but use some type of electrical stimulation to activate certain parts of the brain that's still in circulation today. And all of this was really the precursor to future research that tried to find ways to study the brain in other ways. Another type of thing that we started to develop were kind of tools to build off of some of these original electrical study measures. We could potentially, like we saw in the image in the last slide, hook up sensors to lots of different regions of a person's scalp and just get readouts of kind of the electrical changes that were happening in those regions on a moment-to-moment basis. Or what we could potentially do is combine the signals of all of those different lines and tell us not necessarily about specific activity of the brain, but overall activity of all the different regions of the brain. This led to research on things like levels of consciousness and attentiveness and just overall excitability when we found ourselves in specific situations. Again, us building off of some of the early discoveries that we had about 100 years ago at this point. It also led some really interesting research on how we could really locate not only the specific region of the brain and when it activated, when kind of being triggered by something, but how long that activation occurred. A really popular technique called an ERP, or event-related potential, was designed almost 80 years ago at this point, where researchers would be exposed, or participants, I guess, would be exposed by researchers to a stimulus not just once, but say 100 or 200 times. You can see in this example, this participant had some type of detector hooked up to a certain part of his brain, and then was played a sound. A feedback loop on an EEG of that area, when they're played the sound just once, looks kind of random. It almost looks like the brain doesn't respond at all, really. But if we play that sound, in this case, you see in the example 100 times, and average the height of the readout about 100 times from 100 exposures, something really cool emerges. This ERP that we talked about, event-related potential. And this ERP can tell us, because of how good these sensors are in terms of timing, how long it takes for that area of the brain to be activated when the stimulus is presented, how strong the reaction of that area is to that stimulus, and how long that response lingers. This can give us really cool information about how long it takes for neurons to reset and how long a specific message might linger in certain parts of the brain. It can also, when we combine ERPs with ERPs, give us a sense of a path that messages are taking through the brain when specific stimuli are presented. Again, just building on the conversation that we had, at this point, about a century ago. And this led us down the path of finding other things that we could do, like EEGs or electrophysiology work, that helped us circumvent many of the problems that we discussed earlier. With EEGs, we were able to study kind of basic individuals without waiting for somebody to have a traumatic incident. We also didn't have to break open a person's brain with a lot of the original electrophysiology measures like we did with some of the previous stuff. And we were able to get such large samples that we could be much more confident in a lot of the results we were looking at. And we were able to talk about what was active, not critical or necessary, when we were doing specific functions for the very first time. And this led us on a journey of trying to figure out how else we could study the brain in this exact same manner. But since the EEG and other techniques that take advantage of the action potential started to become more commonplace, other tools started to be developed and perfected by neuroscientists and are also used today. We don't have enough time to talk about the multitude of different things that you might see in a neuroscience lab, but I did want to talk about 3 very common techniques that pop up when trying to study the brain in different ways. One specific technique, called the PET scan, is utilized by taking advantage of another critical process that takes place when cells communicate with each other. Not only do we see that when cells communicate with each other do they give off that electrical pulse that we talked about earlier, but they also, in doing this, lose energy. And the way they regain that energy is to get sustenance from the blood that's flowing through the brain that's crossed what's called the blood-brain barrier. Now, this blood is helping these cells function by providing them with the 2 critical elements that really are needed by all of our cells to continue to function and stay alive. Glucose, which is some form of sugar essentially, and oxygen. If we don't have those ingredients, our neurons can't function, period. So what the PET scan does is not look for electrical changes, but instead they look for where blood is flowing. And the way they do this is by actually injecting a radioactive tracer or isotope that can actually cling to our blood cells, pass through this thing called the blood-brain barrier, and give off some type of emission through a special type of scan. So what people using PET scans do is inject people with these dyes, put them in the scanner, and look for where the blood's flowing, suggesting that wherever the blood's flowing is where there's been activity in the brain. Some of the original PET scans were rather blunt instruments. And we could talk about general areas where there was activity for an extended period of time, but we couldn't pinpoint things very well. Today's isotopes that we use and scanners that we incorporate into this studying have gotten significantly better, down to the point where we can look for specific cells using specific types of neurotransmitters and the paths that are actually incorporated into different types of mental functions. And they provided us with an amazing array of information about different communication channels and processes that are really a part of every mental experience. Another technique that's become very popular, not only in looking at the brain, but looking at a multitude of different body parts, is something called an MRI. Now, MRIs, by design, are essentially depth finders. They let off magnetic pings that can tell us about the density of different parts of our body. You can use this to look for torn tendons, look for structural anomalies in the body, or, when we're talking about studying the brain, look at just how the brain is designed, what the structure of a person's brain is. In fact, one of the cool things you might encounter if you ever get to work in a neuroscience lab or get to actually be a participant in one is you get a nice, pretty image of your brain. As I mentioned, our brains aren't necessarily super pretty all the time, but you can still see your brain through these really cool techniques called MRIs. And if you really want to up the ante, what you could potentially do is participate, once your images have been taken, in something called an fMRI, where researchers inject nothing into you this time, and instead look for some type of neural activity, looking for blood flowing, and utilizing another weird side effect that occurs when blood drops off, this glucose and oxygen that we talked about. When those cells do that, not only have to form an area, but they actually give off a slightly different magnetic resonance when they transition from being oxygenated to deoxygenated. And fMRIs have the capability of locating places down to roughly a millimeter as to where blood had just recently been dropped off within the past fraction of a second. And that can tell us, again, not only where things are active, but in this case, down to a half second or so, when things have been active. And this has given us a very elaborate picture of all the different functions of the brain, and in conjunction with some of the other stuff that we've talked about, told us a lot about activity of the brain. Now, these techniques with the fMRI might seem amazing enough for many people to start to conclude that maybe this is the end of the story. Now, maybe these fMRIs are what all neuroscientists are striving to use today because it's the most perfected thing. But I want to point out that there are some shortcomings with these fMRIs that encourage us to continue to look at different paths of studying the brain. One of the things that's problematic with these fMRIs is even though they can measure things down to a fraction of a second when we talk about functions of the mind is not really sufficient. EEGs or any other type of measurement that utilizes the electrical charge measurements can measure things down to milliseconds. That's what we really need to really pinpoint where things are happening in the brain. Another issue with these particular techniques of fMRIs, and really PET scans as well, is that they're very expensive. And only major research institutes tend to have them at their availability if they want to study the brain. And even then, it's tough to run studies in labs unless there's a lot of funding that goes into keeping and maintaining these tools. And the worst thing with all of these is that even if they can give us amazing images and tell us what's active, they're not capable of doing something that sometimes researchers want to know. And that's telling us about what we need to do specific types of activities. They can tell us what's active, but lesion studies still, for a long period of time, were the only way to detect, to determine what exactly was needed for us to be able to do specific types of functions. Well, until just recently. About 20 to 30 years ago, researchers started to develop one last tool I want to talk about, something called TMS, or transcranial magnetic stimulation research. In these studies, researchers line up two magnetic coils on a specific spot in the brain and start to send magnetic pulses at about 1 pulse per second onto this area, this designated area of the brain. And the individual pulses from each coil passing through the brain have no real effect on the cells that they're hitting. But where they meet, the conjunction of those two spots, over time starts to have effect on the cells. It essentially activates them over and over and over again until they start to become deadened. Not permanently, just for a very short period of time. In fact, the rule of thumb is about every minute that you're stimulating those areas will be knocked out for a minute. But in doing this, researchers have opened some amazing doors in understanding not necessarily what's active when we're doing things, but what's needed, what's critical for us to do specific types of mental functions. And it's created some really fun conversations that neuroscientists are capable of having now that we've developed this tool and gotten better and better at it. Now, for those of you wondering, it is a little painful, like you can see in the image, if you do participate in a TMS study and they start sending these pulses. But it's not painful to the point where you'll be screaming. It's as if you'd be flicked in the head a couple times for, 20, 30 minutes, which is definitely not desirable per se, but not something that will create any permanent damage or really be harmful to you. In fact, your instructor, the person recording this, participated in dozens of TMS studies as a graduate student simply because it was fun, and I was actually paying very well to participate in those studies when I didn't have quite a budget as, what most faculty and other individuals tend to have. So it's another, again, thing that we have as part of our repertoire of studying the brain. And this leads me to the very end of this discussion, and maybe a question that you might be asking yourself. What is then the end point for this? Is there a specific technique that all neuroscientists use? Hopefully at this point you realize the answer to that question is, well, none of the techniques are the end of it, but all of them also serve some purpose. Each technique that we've covered has given us amazing insights into how the brain functions. And when we put the puzzle pieces together from all these techniques, we're able to study the brain in a way that we never could have just 20 or 30 years ago. And it's given people in psychology across a wide range of different areas amazing insights as to how the mind functions. Hopefully, if you're interested in this, you can contribute to this conversation, adding either more information or more tools that we can use at our disposal in our never-ending attempt to study more and more about the brain. This is going to mark the end of this lecture. I hope you enjoyed it. I know this one went a little long, but I promise I'll try to be as concise as possible in future ones. I just know this one is always longer than the rest. But now that we've covered this, it's time to move on to another topic, one that actually is sort of a continuation of what we've been looking at in these past two lectures. I'm going to stick with the biological, but we're going to, as we're talking about biological things, sort of transition to the cognitive side of stuff as we go through this discussion. And this is area that we're going to be covering is, as I mentioned a few lectures ago, really the start of research in psychology, where we started studying something called sensation and perception. We're going to start our next class off by talking about one big process in this formula, sensation, by focusing in on visual processing. There's a little bit that we're going to talk about relating to the other senses, but since we have to narrow things down, that's what we're going to dedicate our attention to in the next lecture. In the following one, we'll be talking about something that's very closely tied to sensation, and that's perception, and the mental breaking down of that information. I'm not going to go too much into it though, because that is for the next lecture. So instead, what I'm going to do, as always, is bid you adieu, wish you all well, and say I hope to see you again. Take care.